chapter 17. Chapter 17 was kind of the oddball of the group. This is where we talk about how we diagnose infections. Now, this one had a lot of different techniques and things in there. I don't expect you to know all of those for the exam. Okay? The biggest take-home message from this chapter is the 3 different groups associated with the tests that we can perform in a clinical microbiological lab. Okay? So phenotypic, genotypic, and immunological. So what I expect you to know is a brief definition of what each of these groups are. Okay? And then the pluses and minuses of each. Okay? So benefits and drawbacks. So we'll start off with phenotypic tests. Remember, phenotypic tests are where you're looking at physically expressed traits. Okay? That can be under the microscope, or it could be, biochemical reactions in a test tube, or something along those lines. Now, the benefits of phenotypic tests are they are the gold standard. They've been around the longest, and we know they're going to work. So if all else fails, that's what you go with. The drawback with phenotypic tests, though, is that they often require culturing. Okay? And when you have to culture, this is going to take a lot of time. So they're time consuming, and that means it can take longer for you to come up with a diagnosis for your patient. Genotypic tests, this is where we look at the genetic material. Okay? So the DNA or the RNA within the organism. The benefit of this approach is that it's very fast, it's very rapid, okay? And the organism does not need to be alive, so you do not necessarily need to culture. Okay? The drawback to this approach is that they are relatively new, so they're not really well understood, at least not yet. And they can be expensive, okay, to perform. Then lastly, the immunological tests. These are where we look at either our patient's sera, so the antibodies that they express, okay? And that can give us an idea of whether or not our patient is infected or has been infected. Or we can use this for identification of an unknown organism by using a known antibody that binds to that organism. The benefit of doing immunological approaches is that they're cheap, relatively quick, and because our patient is basically a walking bag of antibodies, we can do many, many different tests at once. Okay? The drawback to immunological analyses is that oftentimes you cannot tell if your patient is actively infected. Remember, you make antibodies when you get infected, but you carry those antibodies for a long period of time. Okay? So you can't necessarily tell if they are actively infected. All right, so that's pretty much the gist of this chapter. Okay? Again, I don't expect you to know any particular test for the exam, but I do expect you to know the overarching differences between those 3 groups. Okay? So again, phenotypic, looking at physical traits, right? So microscope, biochemical tests, things of those nature. And most of these are going to require culturing of some sort. Genotypic tests, again, looking at genetic material. This is very quick, very easy, however, it can be expensive. And immunological methods are looking at serology or your patient's antibodies to then determine what they are infected with or have been infected with. Blah, blah, blah. And then we went into a lot of details about each test. Again, I don't expect you to know that for the exam. And this just gives you a good idea of why serology is nice, because it's extremely specific, because antibodies only bind to their cognate antigen. And then it's also very sensitive because you have tons of antibodies circulating around the body. Boring, boring, boring. Okay, that's it for chapter 17. So relatively simple. Now let's get into the nuts and bolts of this section. And that is all of the different diseases broken up by body system. Okay? Now, if you've looked at your study guide, which I'm sure most of you have, you'll note that each disease basically has the same couple of questions. Okay? What that's designed to do is get you to jot down just some basic information about each disease. Now, for the vast majority of the diseases we talk about in class, all you're going to need to know for the exam is a brief definition of what it is. Okay? It's a bacterial organism that causes infections in the lung, okay? Or something along those lines. Now, for each chapter, there were, maybe 4 or 5 organisms that we spent more time on, and those are the ones that I'm going to review for this review session. Okay? Those are the ones that may have or may require you to know a little bit more than just a brief 1-sentence definition. But all the other guys that we talked about, okay, the vast majority of them, all you need to know is just what it is and what type of infection does it cause. Okay? So, with that said, let's go into chapter 18, which deals with infections that target the skin and the eyes. Also, for each chapter, it would be useful for you to understand the defenses in that region, as well as if or not there are commensals. Okay? I don't care if you know anything about species or genus of commensals. All I expect you to know is, are they there, and at what level? Okay? So as far as defenses and things go for the skin, the biggest defense is going to be the barrier defenses. This is where you have that keratinized skin layer that protects not only your skin, but the rest of your body from infection. Okay? That's the biggest defense that you're going to find in this area. As far as normal biota or commensals are concerned, they're relatively few and far between, and that's because the skin is dry and inhospitable. Okay? So there are commensals there, but not many. All right, so now on to the diseases. Okay, so this is a prime example of a disease or infectious disease that you only need to know one brief, kind of snippet about. So for acne, I would say it is an infection of a hair follicle with a non-pathogenic bacteria. That's all you would need to know about acne. Okay, nothing crazy. Now we did talk about S. aureus and S. pyrogenes a lot in this particular chapter. Okay. So I do expect you to know about S. aureus and S. pyrogenes and the types of infections they cause, okay? As well as why they are important pathogens. So we'll start off with S. aureus. As we talked about pretty much throughout the rest of the semester, S. aureus can cause disease in pretty much every part of the body, okay? And that's because he produces some pretty important virulence factors. The three biggest ones are going to be coagulase, toxic shock toxin, and protein A. Does anyone remember what coagulase does? Coagulates blood. And why would he want to do that? Protects it from phagocytosis. Good. What about toxic shock toxin? Well, it causes shock. All right? The way it does that is it mimics MHC. So it binds to all your T cells at once. They then get activated, and this results in shock. Okay? Bless you. And then protein A, does anyone remember what protein A does? It binds to antibodies, bringing them out of solution, making them not effective. Okay? S. pyrogenes has similar virulence factors. The biggest take-home message, though, is that they're not as good. Okay? So it tends to be a less severe pathogen compared to S. aureus. So here's an example of an infection caused by S. aureus, furnuncle. This is an infection of a hair follicle with S. aureus. Okay? Again, one sentence definition is all you need to know about it. All right. So one of the bigger guys that we talked about in this chapter was Clostridium perfringens, which causes gas gangrene. Okay? Remember, this is a normal commensal, or excuse me, a normal organism found all over the environment. It's ubiquitous. It is anaerobic, so it can only grow in tissues that have low oxygen. Okay? So remember, this is something that you typically find in people with poor circulation, right? Diabetes, poor blood flow because of genetics and things like that. The organism gets into this low oxygen environment, low oxygen tissue. It then begins to grow and divide, releasing toxins, which slowly destroy the tissue. Okay? This works its way up until you have to amputate and remove large sections of tissue. So biggest take-home message here is that it is ubiquitous, anaerobic, and can only grow in low oxygen tissue. Another big one we talked about in this chapter was chickenpox. Okay? Chickenpox is caused by a virus, varicella zoster virus, VZV. Okay? During its initial infection, it usually goes through the respiratory tract and causes a mild flu-like illness, followed by a red rash that is full of liquid. Okay. This then goes away. However, you're still infected. It's important to note that VZV will hang out in the dorsal root ganglion of nerves around the spinal cord. And then sometimes, potentially later in life, it will wake back up again and then cause shingles. Okay? So please keep in mind that chickenpox and shingles are caused by the same virus, VZV or varicella-zoster virus. Both of these diseases are preventable using the Zostavax vaccine. Boring. We'll talk about that one later. There we go. Next are fungal infections. Okay, we spent some time on these guys. So fungal infections of the skin are relatively common. These occur in people who even have healthy immune systems. And keep in mind that we don't really discuss them or talk about them in terms of their species, but instead we talk about where they actually infect. And remember that the fancy name for fungal infections of the skin is tinea, and depending on the area of skin, it gets a different name. Okay, so tinea corporis is smooth skin, tinea pedis in the foot, tinea cabatus in the scalp, tinea barbae in the beard, tinea gudum in the nails, and tinea crucis in the groin. All right, on to the eye. So really, there's only two areas that can get infected in the eye. That is the conjunctiva and the cornea. Okay? If you get an infection in the conjunctiva, this causes conjunctivitis or inflammation of the conjunctiva. In everyone but neonates, this is harmless and goes away on its own most of the time. However, in neonates or newly born children, it can cause significant scarring and potential blindness. And for the cornea, really it's just having a long-term infection that causes scarring of the cornea. Okay, relatively rare. And then lastly in the eye, we have onchochoriasis, okay, also known as river blindness. Remember, this is a helminth infection that can detach the retina. Okay, that's it for chapter 18. Now on to chapter 19. Nervous system. Okay. As far as the defenses go in the nervous system, in particular the central nervous system, the best defense present is the blockade of peripheral things getting into the body or into the central nervous system. So it's having a completely separate, closed system. Remember though that this is a double-edged sword. Because if you do happen to get an infection in this closed system, then there's nowhere for the inflammation and the edema to go except put pressure on the brain, and this can cause brain damage. And as far as normal biota goes, there should be absolutely none. Okay? No organism whatsoever. All right, so the first group of illnesses or infectious diseases that we talked about is meningitis. So remember meningitis, this is inflammation of the meninges, and in this case this is due to an infection. So a general rule with all meningitises is that bacterial meningitises tend to be the most severe. These often result in 80–90% mortality. Where viral meningitises are not as life-threatening, they only have about a 1% mortality, but they still cause brain damage. Now, out of all the meningitises, the one in particular that I think you should know a little bit more about is Neisseria meningitidis. Okay. This is a Neisseria organism, so bacteria, and this organism can get in through the oral cavity, okay, so through contaminated saliva. And so for that reason, it can cause community-acquired meningitis, okay, or transmissible meningitis. This organism, when it gets into the brain, it then attaches to the meninges, causing inflammation, and can cause a life-threatening bacterial meningitis. Okay? And that's true with most bacterial meningitis. Now, for all of the other bacterial meningitises, I don't really care if you know any particular organisms by name. However, you should know that for all of the other bacterial meningitises, you often have to be slightly immunocompromised. Okay? And remember, that's because they usually start in the lungs, work their way into the bloodstream, and then get up into the brain through the blood-brain barrier. And for that to occur, your immune system has to kind of ignore it or be severely weakened. Remember, all bacterial meningitises are severe, usually resulting in about an 80 to 90% mortality rate. We also talked about some fungal meningitises. One of interest is Coccidioides immitis, and that's because this guy can get into even healthy individuals. Remember, it often starts in the lungs and then works its way into the brain and can cause severe meningitis, even in healthy people. And then again, viral meningitises are less severe in the fact that they do not cause mortality that much. However, they do still cause brain damage or neurological sequelae. Next, we have meningoencephalitises. And this is where you have